Yes, greetings, my students and everyone who is connected to Metric Hub. I welcome you to 2025 academic year, and I'm quite sure that we have got long-time partners, long-time students, long-time individuals who have connected to the channel. 2025 is come, and we are gonna do some better big things again. Don't forget to subscribe. I'm sure that from the previous years, you have seen and you have got testimonies of those who have worked with Metric Hub. This is Great Cove Life Sciences. I'm coming to you with the first presentation of the year, and I'm sure that you will never have any problems and you are going to enjoy the presentations and all other lectures to come. So I'm going to be presenting to you life at a molecular level, the concept and the ideas, everything to do with DNA structure. So you can see this displayed on the body where we have got the nucleotides, we have got the nucleotide, we have the base pairing, and we have got the double strand or the double helix structure of the DNA. For those who have done life science before, for those who have met and revised, gone through some previous question papers, I'm sure you are able to join the dots on what is drawn on the board right now without wasting your time. This is what we are going to discuss about in this presentation, everything well explained and articulated to. Let me just quickly go through my presentation, which I'm going to be dealing with you today. Life at a molecular level, the DNA. What is DNA? I'm sure that DNA you have seen or you have heard that it's an abbreviation. It is an abbreviation which stands for deoxyribonucleic acid. Is deoxyribonucleic acid, commonly known as DNA, which is actually a complex molecule that contains all the information necessary to build and maintain organisms. I'm sure that we have seen or we have heard and we have revised and passed through uh, presentations, information explaining about the DNA, which we are to agree that it contains the genetic code. It is the one that influences the structure, the appearance, and the functioning of every living organisms. So I'm sure that we have also heard and we have also seen that all living things have DNA in their in their cells. Living organisms have got deoxyribonucleic acid in their cells. So almost every cell in a multicellular organism possesses the full set of DNA required for that organism. So the functionality, the existence, the living of any organism is controlled as a result of what? Of a set of DNA that is within that organism. So, however, DNA does more than specify the structure and functioning of living things. It also serves as the primary unit of heredity. DNA also does more than only controlling the structure and the function of living organisms. It also serves as the primary is to say the key unit that affects heredity in organisms of all types. How we are saying whenever organisms reproduce, a portion of their DNA is passed along to their offspring. The ability of organisms to reproduce young ones or offsprings that are more or less similar to them is because of heredity, it's because of inheritance. That is an idea that I want you to understand. That information is passed from one generation to another. Characteristics of organisms, features of organisms, functioning of organisms, the appearances of organisms, everything that we can define with regard to living organisms, it is controlled by DNA. It is DNA that actually affects that, which we are saying it affects the inheritance. It affects the heredity in what in passage or transmission of characteristics of organisms from one generation to the other. Nucleic acids are composed of nucleotides. I'm sure and I'm quite aware that from your introduction of your classes this year, those who are starting grade 12 this year, I'm sure that we have heard of nucleotides, which we said are the building blocks of what? Of the DNA from the diagram that I was presenting on the board during my setup of the class, or when I was just displaying the structures and diagrams that I was going to use in this presentation. Uh, you can understand and believe with me that we have got sugar, we have got phosphate and nitrogen base, which we are saying these are the components of what? Of nucleotides. So what I'm sure of, or what I want you to understand, is that DNA is made up of nucleotides. I'm just giving you the introduction. We'll go deeper into the structure as we go through the presentation. So nucleotides, they also contain what? They contain deoxyribose as sugar. So the DNA has got a sugar which is known as what? As deoxyribose. Deoxyribose is the sugar that is found in DNA. I'm sure you need to quite take note of that, and make sure that you don't make mistakes. Note it down that DNA has got a sugar that is found in it, which is found, or which is carried on the nucleotides, which is also known as the deoxyribose sugar. So I'm sure that you are going to make sense of it as we go. And then the functions of DNA, the functions of DNA. The primary function of DNA is to store and transmit genetic information from one generation to the next. I'm quite aware, and we have wondered, some of you, that how is it possible that parents, they give birth children who look alike to them, or children who are born from the same biological parents, they look alike, or more or less, if they are identical twins, they are exactly the same. What I want you to understand in this presentation of introduction to genetics, or introduction to this great core of life sciences, I'm sure that you need to know, and you need to take note of the concept of DNA as the molecule that transmits, that stores the genetic information from one generation to the next. It is done by DNA. It serves as a template for the synthesis of RNA. The synthesis of RNA, which then which therefore directs protein synthesis through translation. So in terms of the synthesis, we are talking about the formation of RNA. The formation of RNA, which I'm sure that you need to also take note of, is what? mRNA, which is formed. When is that formed? During what? During transcription. Formation of RNA takes place during transcription, when we are to see that DNA, it actually produces, it makes a single strand from itself, which is formed during the process of which is formed during the process of what? Transcription. It is transcription. We are saying DNA information is being transcribed, it is being transferred from the DNA molecule into an RNA structure, which we are saying it's a single strand. So the synthesis, it serves as a template for the synthesis. In order for the RNA to be formed, mRNA to be formed, it is formed because we have a template which complements to the free floating nucleotide bases that are found in the nucleoplasm of a cell, which therefore forms a single strand that we call mRNA. So please note that it serves as a template in that process of what? Of transcription. DNA also plays a crucial role in DNA replication during cell division. The idea and the concept of DNA replication during cell division, ensuring accurate duplication of genetic material. When cell division is to take place, replication is to occur first, which is the ability of what? Of DNA to produce multiple copies of itself. These copies are produced or are formed as a result of what? Of the duplication process, which I'm saying it plays that key role that is done as a result of what? Of the multiplication of the DNA molecules, which we are saying it is therefore the DNA has to be more or it has to be enough in order to ensure the duplication of genetic material. Again, the next point, it carries the genetic code that determines an organism's traits, characteristics, and inherited features. DNA carries the genetic code. It carries the genetic code that determines an organism's traits. We are talking about characteristics, features, and also the inherited characteristics or inherited features. Organisms do inherit or they do get the features of their parents, which is transcribed into them, which is carried by what? By the gametes, the sperm cells or the egg cells. So in that respect, what I want you to understand is that the sperms and egg cells in living organisms, they have got the capacity of carrying the genetic code to the next generation, which is carried within the chromosomes or carried in chromosomes, which are in those, which is carried in those what? In those sex, uh, which we call the sex cells. So I'm sure we are still together there. And let's look onto the structure of DNA. The structure of DNA. The structure of DNA. It is made of two strands that winds around each other to resemble a twisted ladder. It resembles a twisted ladder. The structure of DNA, it is like that. It is the DNA structure. It is actually formed by two strands that twist around each other, resembling a twisted ladder, which we call the double helix. If you can check in previous or various papers as we prepare and get ready for our any exams to come, the DNA structure is known as a double helix structure. That is the scientific representation or structure and appearance of DNA. It is what? It is DNA structure is a double helix. It is a double helix which has got the nitrogen bases that are sustained or subtended by what? By weak hydrogen bonds. So we are saying these, which you are seeing on the body here, we are saying this is called the what? The strands. These are the strands that we are talking about, which you can see that they are twisting along each other or around each other. So therefore, you can see that they are twisted and resembling a ladder, but however, it is twisted. Each strand has a backbone made of each strand. Each strand, it has a backbone made of alternating sugar, deoxyribose, and phosphate groups. So what I want you to understand is that as you look on this structure, we are saying uh, each strand like this, we are saying it is made of what? Phosphate sugar molecule. There is phosphate and what? And sugar. Phosphate and sugar molecules are the ones that are forming each of the strands that you are seeing there. They are the ones that forms the what? The strand that you are seeing. And then inside, of course, we have got the what? We are saying inside the strands, we have got nitrogen bases. I'm going to present to you on the board here as I go through the presentation. You will see this. I have got clearer diagrams that are attached below there. Just keep following. And then I'm sure again that you need to, I've already talked about the deoxyribose, which is the sugar that I said that it is found what? It is found in DNA. The deoxyribose sugar is that sugar that is found in DNA, and then attached to each sugar is one of the four nitrogen bases, or is one of the nitrogen bases, which is one, the adenine, two, we have got the cytosine, and then we have got the what? We have got the guanine, which is three, and then four, the what? The thymine. So what happens here is that you need to understand as well that when we are into the types of nitrogen bases, there are four types of them. There are four basically of them, which you need to know again that as we go with the subject, we are not going to see where it is written in full to say that adenine cytosine, but you identify that they are written with what? With abbreviations or initials that are given there. So A stands for adenine, C for cytosine, G for guanine, and also there is a T that is for thymine. So DNA has got four nitrogen bases. DNA, it has got four nitrogen bases. It has got four nitrogen bases, which I said it is A, adenine, it is A, adenine, it is C, thymine, it is G, guanine, and it is T, which is thymine. So what I'm saying is these are the four nitrogen bases that are found in what? On the DNA structure. So as you can see here on the board, I've just shown you that I'm just showing to you that what happens is that we have got the purine and the pyrimidine bases. That one other area which I'm quite aware that most of the students and many of the learners are not aware. Why do we have the complementary pairing, or why do we talk about the complementary pairing? Why do we talk about that? So what I'm saying is that you can see here, total purine is equal to total pyrimidine basis. What we are saying, or what you need to know, is that A and adenine, it pairs with thymine. This equal sign that you are seeing here, the equal sign that you are seeing, this one, it is just showing that it is a complementary pairing. It is showing that this is the complementary, it's a complementary pairing taking place where A and T pairs together and G and C pairs together. So what happens is that the purine basis and the pyrimidine basis, they are supposed to be understood to say that they are always equal to each other. So we are saying the total number of purine is equal to the total number of what? Of pyrimidine bases. Why is that so? It is because of the complementary pairing, which I'm going to explain as I go with this presentation right now. So I'm sure you can see we are saying G and C, which means G, it pairs with what? With the C, as you can see here. G is equals to C, which means G pairs up with what? With the C. So what is purine? What is purine and pyrimidine bases? Basically, these are nitrogen bases that makes up the two different nucleic acids, the DNA and RNA. The purine and pyrimidine bases are nitrogen bases that are there to make up the nucleic acids. I'm sure again that you know that the nucleic acid that we are talking about is what? Is the DNA and RNA, which are the two nucleic acids that we need to take note of. I'm saying they form the what? They form the DNA and RNA nitrogen bases that you can see there. So, having said that, having said this, just take here now. I'm sure like I've said the purine bases, which are those ones. The purine bases, we have got what? The adenine and guanine. The purine bases we have got the adenine and guanine are two carbon nitrogen bases. They have got two carbon nitrogen ring bases that are there. This one, purine bases, is A and G. I'm saying this is A and it's A and G. They are purine bases. So pyrimidine basis is what? Is cytosine and thymine are one carbon ring are nitrogen bases. So sometimes you know that our syllabus has been tested quite a long time, and it has been repeatedly done and and tested. So please, what I want you to know is that we cannot sometimes just easily ignore things. I want you to know that the purine basis is adenine and guanine, and then the pyrimidine basis is cytosine and thymine. And that is when you see again that one class is two carbon nitrogen ring basis, and the pyrimidine basis is what? Is one carbon ring nitrogen basis, which you can see. And then, as you can see, we have said again that there is a base pairing whereby to say complementary. Complementary, therefore, is explaining that we cannot have preline bases pairing on their own. You'll see that we have got A here, it pairs with what? With the T, which is on the pre-methane basis, and then we have got also G, which pairs with what? With the C here. So what is important is for you to understand that complementary pairing, it is happening because of what? Because of the two classes that are joining together. As you can see that A, it joins with C, A joins with T, G joins with C there. So that is it. There are three main types of pre-methane bases. However, only one of them exists in both what? In both RNA and DNA. So that is what? That is the cytosine. Cytosine is an example of the pre-methane bases, as you can see in the on the board as represented there. Cytosine is a pre-methane base, and then we are saying there are three types of them. The other two are uracil, which is RNA exclusive. We know that uracil, it is also a pre-methane base, and it is actually only found in RNA. And then thymine also, it is also DNA exclusive, meaning to say that it's only found in what? It is only found in DNA. So what I want you to understand is that the pre-methane bases, there are three types of them, which is uracil, thymine, and what? And cytosine. They are the three types of what? Those are the three main pre-methane bases that are there. Each strand has a backbone that is made up of phosphate sugar chain, which I have already explained there that there is a phosphate sugar chain that is making each of the strands of the DNA structure. Attached to each of these sugars is a nitrogen base that is composed of one carbon and nitrogen rings. It is therefore that you need to know that there is what they are nitrogen bases that are also forming the strands or connecting the two strands. Having said that, we need to understand again the number of rings this base has determined whether the base is preline, which is two rings. I have already said so that the number of rings this basis here defines or determines whether the base is a preline, which is two rings, or pre-methane, which is one ring. So the pyranine on one strand of DNA forms hydrogen bonds with the corresponding pre-methane on the opposite strand of the DNA, and vice versa, to hold the two strands together. So in other words, what we are saying is that we can have our strand like this strand there. One, we can say strand one, and then we have got another strand there, which we can say strand two. So what I'm saying is that we have got one strand here, which is to say that, for example, we are saying it is the purine, which is on the other side, we have got one other strand there, and then we have got this base here and that base there. So in other words, I'm saying it is A here, and then it is T there, it is C here, and it is G there. And in between them, there is what? There is weak hydrogen bonds that joins the two bases together, or that joins the two strands, the complementary strands, or the complementary bases to join together. So what happens is that is they form, we are saying they are corresponding pre-metals on the opposite side of each other, or vice versa, to hold the two strands together. Within DNA molecules, this is the most important function and most and well-known reason why we talk about, or which is known as complementary base pairing. The complementary base pairing, which is to say A, which is from the purine, is pairing with T, which is from the pre-methane basis. C, it is from the purine, is from the pre-methane is pairing with the G from the purine basis. So within these DNA molecules, it is most known that it must be known that bases always complementarily pair. There is complementary pairing. There is complementary pairing, which is explained by one class of DNA. One class of nitrogen bases pairing to the other class of nitrogen bases is according to the classes that I have just posted there. And there we are able to explain and answer another question that comes on the multiple choice, which you guys, you must understand that the ratio of two will always be constant within a DNA molecule. If we have got, for example, the ratio of two is always constant, it means to say that, let me assume to say that uh, we have got 15 A here. It means T is also going to be what? Is going to be 15 as well. So what we are saying is the ratio of the two will always be, is always constant within the DNA molecule, is to say that the bases that pairs to each other have equal numbers. 15A is equals to 15T. 20G is equals to 20 what? 20G. We are saying here we have got 20. It means we also have 20 what? 20G there. So this is the constant numbering or the constant ratio which I'm talking about. In other words, one strand of DNA will always be an exact complementary of the other as far as the purine and the premethine bases go. So as long as we are talking about this, we cannot have one strand being longer than the other because these bases, they always have equal numbers as according to their pairing. Hence, we are saying the strand or the DNA molecule, it will have what? It will have a proper and specific shape. So look here again. At 9 pairs with what? With thymine, as you can see, and two hydrogen bonds. I have already illustrated about that. I'm trying to explain as much information as I can and give you precision with regard to DNA structure. And then guanine pairs with what? With the cytosine, and three, it has got with three hydrogen what? Hydrogen bonds. What happens is that I can give, for example, to say in terms of A, you see that this is A there, the two hydrogen bonds that are talked about is like this. And then if it is G and C, we are saying there are three uh, hydrogen bonds that are found there. The three hydrogen bonds are these ones. These are called weak hydrogen bonds or hydrogen bonds that we are talking about. As you can see on this on the first diagram there, on A2, AN is 2, and then on the other diagram here is 3. I'm talking about the red lines that are joining A and T. So A, T means A pairs to T, and we have got 3, we have got 2 weak hydrogen bonds that are joining there. And GC, we have got 3, which you can see those 3 lines that are connecting C and G there, which are 3. So NB, I'm saying take note that one way to remember which bases go together is to look at the shapes of the letters themselves. Look at the shape of the letters themselves in order for you to understand the base pairing. I'm saying one way to take note or to remember this, one way to remember this, and B, I'm saying one way to remember it is for you to look at the shape of the letters that are look at the shapes of the letters that are connected or that are joining. So as you can see, the letters made of only straight lines A and T are paired with each other. Class, take note of this. Take note of this. Letters that are made only with straight lines, A, T, are paired with each other, while the letters that are made up of curves, G and C, also, they go together. This is quite nice and is so typical to, for any learner and easy to reflect and remember. Letters that are made of straight lines, they join together. Letters that are made up of curves, C and G, also, they do likewise. So because hydrogen bonds are not as strong as the covalent bonds, base pairing can easily be separated, allowing for processes of what? DNA replication. They do allow for processes of what? the replication process, and also transcription, as we have mentioned in the earlier, as I alluded in my introduction above there. So weak hydrogen bonds, why are the complementary bases joined together by weak hydrogen bonds? It is for this reason that the weak hydrogen bonds must allow for DNA replication as well as transcription process, where the single strands, where the DNA must unwind, and then also the weak hydrogen bonds blow, they break off, allowing uh, the basis to play from the free floating nucleotides that will form and there and there. We are not to explain about DNA replication, but I just want you to understand that weak hydrogen bonds are weak to allow for processes of separation that must take place during DNA replication and also transcription. So concluding on my presentation is the nucleotides as the building blocks of the DNA, as I have just explained to you. The certain times in our question papers, we find that there is this structure which is made up of the three molecules here, which is, I'm saying, one, two, and then three. So these ones are the ones that are forming. I can say structure A is the one that is forming structure B, and then structure B is equals to structure what? Is equals to structure C. A forms A forms B. It actually forms B, and B is the same. B forms B, which is the same as what? As C. It forms B. A forms B. A, it forms B, and B is equals to C. B and C is the same thing. B and C is the same thing. So what is happening here is that we are having our structure B. Let's start with, let me just start with A. Let me start with A, yes. A is the nucleotide structure, which is forming the whole structure B. So as you can see, we have got phosphate sugar backbones that are shown on the diagram here. The phosphate sugar bonds, the phosphate sugar backbones here, we said that the strands of DNA, this strand, which I can just maybe shade to say for you to see, I'm just highlighting this for you to see what I'm explaining about. This is the backbone which we are talking about. This backbone, it is made up of what? The highlighted, the one that I'm putting the red marks there. It is the phosphate sugar backbone, which is molecule 1 and 2. Molecule 1 and 2 forms the strands. Phosphate sugar forms the strands. They form they form the strands of DNA structure. They form the strands of the DNA structure. I'm saying 1 and 2, they form the strands of DNA structure. And also 3, it is forming, as you can see, we are saying it is A, T, C, G, adenine, thymine, cytosine, guanine. These are the ones that you are seeing. It is A, which you are seeing here, that we have got A, we have got T, we have got C, G, continuously going down like that. These are the nitrogen bases, which we say that DNA has got 4 nitrogen bases. We have got the four bases that you can see there. The 4 bases that you can see here. The 4 bases which you can see. The 4 bases that you can see there. We are saying A, T, C, G are those ones. So this whole structure, as you can see, the arrow here, it is showing just a segment of the structure of DNA, explaining to say that it is formed or it is made up of what? Of the nucleotides. So here again, understand the 3 nitrogen, the weak hydrogen bonds that we talked about, to say GC has got 3, and then AC has got 2. I'm sure you see here we have got 2. We have got 3 here. We have got 3 there as well. We have got 2. We have got 2. We have got 3. We have got 3. So what I'm saying is that those numbers that are at the center, which I'm putting digits, which I'm number, I mean these dots, which I'm putting numbers there, we are called these. These are called weak. They are the weak hydrogen bonds. The weak hydrogen bonds that we talked about, these are the weak hydrogen bonds. The structure that we are looking at right now, the structure at B, we are saying this is a structure which when DNA, or which is assumed by DNA during the process, or when uh, DNA replication, or it has to, or we are going to talk about replication, or we are going to talk about a small segment of DNA unwinding. That is the unwinded structure. And then this one is the double helix, which is the double helix structure, which is the original state where or in which DNA is supposed to be in. So the complementary coloring that you are seeing on this structure here, on structure C, the structure C that you are seeing here, it is just trying to illustrate that it's different nitrogen bases that are coming up together here. They are just giving you to say that you need to know that these are different nitrogen bases that are joining. For example, if we are going to say we have got C here, if we say there is C there, and then sometimes they want you to find what? To find this one, which we can say it is Y. Identify what you call nitrogen base Y. Nitrogen base Y, how do you know what is Y? You know Y according to the complementary pairing, to say Y must be what? Y must be G, because C and G, they pair together. If we are going to say that this is T, then it means on the opposite side we have got A. So please, class, I'm sure that we have heard, and this topic is going to make the primary foundation of your understanding towards everything to do with genetics and inheritance. It is quite important for you that you take note of it, and you are not gonna miss and forget it. Keep on revising and reflecting to it, so that when it comes to exams, all of us, we are going to write with a smile on our face. Uh, Thank you so much for your time, and I'm quite happy that it is our first lecture, it is our first presentation, and more are still to come. Don't forget to subscribe, to share, and also to refer friends and family. Metric Hub is there for us. It is going to ensure that we are together until our academic success is achieved. All the best. Keep connected, keep subscribing, and turn on your notification button in order for you to take note of new and additional presentations that are coming. We also have presentations on other lower grades. They are as well coming. Bring your brother, your sister, grade 8, 9, 10, and 11. They are also here. Please take note of that. All the best, and have your beautiful study, and have a happy academic 2025. Thank you.